I mentioned in a recent video that WGS tests, these are whole genome sequence tests, are increasingly adding new branches to all the haplogroups on the Y-fold tree, because the Y-fold tree accepts samples that are tested from all vendors as long as it's the kind of test that tests the entire Y chromosome, or some some tests cover different percentages of the Y chromosome. WGS tests are able to cover nearly the entire Y chromosome. And so there are some regions that are just not covered by big Y. And so these branches will never be on a family tree DNA only tree. And now we're seeing a difference between the Y-fold tree and the family tree DNA version of the tree for my haplogroup, J2B L283, and the child Z1043. This is one of the more prolific groups. Uh, Back when I got into Y-DNA in 2015, oh, that's when I ordered Geno 2.0 in 2015. But the results took some time, and then I started to get into it gradually, more and more and more, as my curiosity got piqued, and I learned where my relatives were from all over the place. Back in the day, many years ago, there were three branches of Z1043, J-Z1043. One of them was J-Y26712. The Wardens of England are maybe the most well known of them, because early on they were kind of pioneers in Y-DNA testing. Not so many of them did the advanced tests, but they did STR tests, and then one or two of them did a big Y, established their branch, and some other guys. I'm I guess there's not so many, so I can mention them all. Someone from the Azores, a guy from Switzerland, and then there's a branch that has some Germans from Rhineland-Pfalz, and then a more Eastern European distribution branch. But all those guys in the Eastern European distribution branch descend from a guy from 1500 years ago. So this was one of the first branches known. The other branch that was known was at the time called PH 1553. There are so many dozens and dozens of guys in this branch who have bought STR tests in the beginning. Now they've done the more expensive tests, the big Y and whole genome sequence tests. Actually, I don't think any of these guys did a whole genome sequence test yet. But they're probably going to now, because I've recommended them that we can find SNPs that aren't found by big Y, and then get more branching points. Why do we want more branching points? More branching points can resolve, is this line really more closely related to this other one or not? Um, it has implications for the phylogeography of, well, if this line is more closely related to this one, then they're looking at those two, their common ancestors. Geography may have something to do with where each of those two children line wound up. Okay, the third line of Z1043 that had been known from the very early ages of the testing was Z8424. This is the most prolific one, and there are so many guys in here. There's me, Belgium. We got this Viking from 1,000 years ago on the island Kalmar, I guess is the name of the island, of Sweden. There is a Jewish branch of the many dozens and dozens of Roman era branches. One of them are Jews today. And we've just got guys from all over Europe, and we got Tatars and Moksha, people that are really extreme geographic outliers from everybody else in this lineage. So it's an interesting. We're interesting because we've got like every single kind of people in Europe, and even people outside of Europe. And look, there's even a guy from Syria. His male line comes from Aleppo. Uh, he's the only guy so far in Z1043 that's from the Middle East, to my knowledge. Okay, so the three oldest branches that were first discovered of Z1043, I just ran them down. There was last year, I think, in the summer, a breakthrough that a bunch of guys that were not PH 1553 ended up sharing a SNP that united all of them together. And this was a very big deal. This was like the biggest, in my opinion, the biggest breakthrough in J-Z1043 research was this SNP, FGC 55778. This SNP was discovered by Full Genomes Corporation. We know that because it starts with FGC. So I guess it was one of their whole genome kind of products. But Family Tree DNA added this SNP to their phylogeny, and so did YFOL. And they have coverage. Big Y's and WGS tests both have coverage for this SNP. And it was an indel. It was a deletion. And these kinds of SNPs are harder to analyze. And I think that's why it took many years until finally Family Tree DNA and YFOL recognized that this SNP unites all these guys. In their case, all these guys had a very distinctive STR allele. DYS385 is 10. This was a deletion in the allele from a 13 or a 14, which most everybody else in our line has. So it was not a shock to find that all those different lines were related, because they had this like really weird mutation in the STR. And finally, it was proven by a SNP test that, yeah, that was indicative of just one ancestor had that mutation. He had that STR mutation that was very drastic, and he had a SNP, FGC 55778. Okay, that was just a little bit of backstory. Now, fast forward to today. I believe, oh, this is not the live version. Okay, we got to go to the live version of the tree now to see the breakthrough. Now, I think now is the biggest breakthrough since this FGC 55778 united all those other children into one line. Today, it's an even bigger breakthrough because the two lines that are now being united are the two of the oldest lines of Z1043 that nobody knew were actually descended from a common ancestor. So what we found, Y26712 and Z8424 actually descend from Z8427. This is significant because over 2/3 of all the guys in Z1043 are under one of these two branches. So here's a little tree that I made showing you what this means. This is on YFOL version 905 live version. Z8427 has these two children, Z8424 and Y26712. Okay, before I'm going to explain this next part, it's a little bit more complex, but I'll just show why did we find this on the YFOL tree. We found it because, oh, well, let's look at the family tree DNA tree. So I was curious, why doesn't this show up on the family tree DNA tree? They've got Z8427 several branches up. They've got it at Z631. So I wanted to check, is that accurate? And I wasn't able to find any family tree DNA big Y samples that have any coverage for Z8427. I didn't check every single one, but like I checked, 10 or so, and none of them have any coverage for Z8427. But I was able to check that whole genome sequence tests do have coverage for it. And this guy from Italy, Treviso, he's in a branch of Z631. He's negative for Z8427. He's got reads and they're negative. And I also was able to see this Slovenian ancestral surname Manfreda. We helped them buy a WGS 400 test at YSeq. He has a read. It's negative for Z8427. So it doesn't look like it actually was inherited from Z631. J-Z631, because those guys are negative. We find it positive. First, Weifel had it defining Y26712 because the sample from Russia is positive for Z8427. And then, as part of their review, some months later, after me having upgraded this Bulgarian guy, we bought a WGS test from YSeq. He was one of the first guys that I upgraded with funds from donations from our project members. He was one of the first guys we upgraded, and he ends up being positive as well. And I guess just Weifel didn't notice it at the time, but they found it now, and because he's positive, and because this guy from Russia is positive in these two different branches, and because all the other guys that they have reads for are upstream are negative, Weifel put Z8427 just above Y26712 because that's what makes sense from the reads that they have. So now this is what it looks like. Now the more complicated thing that is interesting is maybe we can finally resolve whether or not Z8425 is a valid SNP to use in the phylogeny of J-Z1043. This has been on the family tree DNA tree for several years, and family tree DNA tree says Z8425 is the parent of Z8424 and BY38004. Weifel decided it's not reliable enough SNP because it could represent many different regions within the Y chromosome, and you don't really know which one when your software aligns the reads together. You don't really know where it came from. It could have come from like a dozen different locations on the Y chromosome. So I think that's roughly the reason why they decided not to use it. It's a homologous region. Some homologous regions the Weifel adds to the tree. This one they didn't. It still could be valid. It's because I was interested in this, and I found that one of the samples that Family Tree DNA tested, they said that he was Z8425, but then we found out later he definitely couldn't have been because he's FGC 55778. And so then they fixed that, and then what they told me was this SNP before HG38, could not be reliably read. But they are confident that HG38 it can be reliably read now. Well, we are now in an interesting position that we can verify this with another piece of information. Because Z8427 is reliably read by whole genome sequence tests, we know that J-Z8424 guys are positive for Z8427. Putting these two pieces of information together, if Weifel is right and if Family Tree DNA is right about Z8425, that we should accept it, then this is what the tree looks like now. If you put both of this information together, kind of like merge these trees, this is the only tree that would make sense. because I put a little note here. This sample from Russia that is Y26712, he's negative for Z8425. So Z8425 could not possibly be upstream because he's negative. So it's got to be, if it's correct, then it would be above Z8424 and below Z8427. So it's where you see in that tree. Um, so what we can do, and what I hope that we're able to do, because if Z8425, I couldn't find a contradiction in any sample that shows that it's wrong to use it. If it passes this test, if we do a WGS test for any guy in JBY380004 or Vetter of North Holland descent, when the guy's related to him, he's got a bunch of guys related to him that have like a smoking gun STR allele signature, that they have such rare alleles that they got to be each other's closest relatives. Unfortunately, most of those guys were not very they didn't want to do the expensive test yet, like the Big Y or a WGS. But now I've got to recommend them to do a WGS test because I want coverage for Z8427. If either of these guys are positive for Z8427, then it's some additional circumstantial evidence that shows, okay, maybe we should accept this SNP that Family Tree DNA said puts on their tree because it's borne out in this phylogeny that they are downstream of Z8427. But it could be disproven if they are negative. If they are negative for Z8427, then there's a contradiction between the trees, and then either Z8425 should be thrown out for phylogeny, or Z8427 should be thrown out. But Family Tree DNA has both of these SNPs in their tree, but where they have it is contradicted by the WGS samples that I've looked at that have coverage. WIFL only has one of these SNPs in their tree. They are of the opinion Z8427 is more reliable, and they don't have Z8425. So I think they had it in a very early version of the tree. So anyway, I'm going to recommend here, if any of you guys are seeing this and you're in JBY38004, most of these guys are from British Isles. There's the guy from Serbia with ethnic German ancestral surname, Schweitzer. And there's guys from England and Vetter of North Holland. Recommend these guys to do WGS, and then we will get some better information to know how to handle this. Does this breakthrough? It's a breakthrough because we're uniting two branches that for years, for like a decade or however long that these branches have been known, nobody knew that they were actually each other's closest relatives. I didn't yet find any STRs among the 111 that show that we would expect this. Maybe if I look closer, I can find it. But does this breakthrough answer the big question of where did these guys come from? That answer is no. That doesn't answer that question. Doesn't answer that question now. Both of these lineages are extremely geographically diverse, and they do not have ancient samples dating back to the Iron Age that we would need in order to conclusively know beyond the shadow of a doubt where they came from. But in the future, we will have more samples, and we will eventually have some Iron Age samples in our group. We're so prolific; it's gotta be found. So it will be important that we get this phylogeny right, because then we can make a better inference based on who is related to each other and where they are from. Thanks very much.